Hi, everyone. I'm very glad to introduce our last speaker, Dr. Patrick Shih. Dr. Patrick Shih got bachelor degree in UC San Diego and a PhD in plant biology in UC Berkeley. After working as a postdoc in Stanford University and the Lauren Berkeley National Lab, he started to work in UC Davis as assistant professor in 2018. Patrick right now is working in the Department of Plant and Microbial Biology in UC Berkeley. Patrick got a lot of awards and mentored many graduate students and the postdoc. And he also actively involved in many academic activity. So Dr. Shih is right now doing excellent research on engineering metabolic pathways using synthetic biology method. So please join me, welcome Dr. Patrick.
Okay, thanks for the invitation. Everyone can hear me okay? I'm assuming the sounds means yes?
Yeah, we can hear you.
Okay, cool. That sounds great. Okay, so this has been a super cool symposium. Thank you for the invitation. I'm really sorry I couldn't be there in person. I can't remember the last time I was at like a meeting or a symposium that had so many different topics. So I think we're gonna take quite the turn in research and switch over to plants and think more about metabolism with my talk. Pretty much my group is located at UC Berkeley, but half of my group is also funded by the Department of Energy Bioenergy Research Center called JBay, or the Joint Bioenergy Institute. And really what kind of brings together my group, both at Berkeley and at JBay, is kind of this interest across both parts of the lab in redesigning and using synthetic biology to engineer plant metabolism. So that'll be some of the stuff I'll be talking about today. Okay, so I just want to start off by first framing kind of my perspective on where the field of synthetic biology is, not just in plant biology, but overall. And really, I would say synthetic biology has been around for arguably over 20 years now, and people really focused on model microbial systems like E. coli and yeast. And really, we've been able to engineer really cool genetic circuits, engineer complex metabolic pathways, and it's been really cool to see how the field has progressed over the last 2 decades or so. But I think the field in the last 5 or so years has really gotten much more interested in realizing that there are huge limitations in working in just these model microbial systems, where you have to essentially, if you want to use them for some sort of applied research or like transition them into industry, you're pretty much throwing them into some sort of large fermentative tank and making potentially some sort of product. And so my group's been very interested in how we can take a lot of the principles and lessons that we've learned in these synbio lessons from microbes and start to transition them into plants. And so I'd argue that plants offer a really unique platform to begin doing some cool metabolic engineering or synthetic biology, because one, plants are photosynthetic. You don't have to add sugar to make them make things. All you need to do is get some seed, throw them in the soil, give them water, give them sunlight, and they just grow and produce many, many different compounds. The other reason we're really excited about engineering plants is because you can begin to start reimagining the scale at which we do synthetic biology. So instead of, again, going back to this, hopefully you guys can see, let me get laser pointer going. You know, instead of just going for these engineered microbes that then have to be dumped into fermentative tanks, we can begin to think of engineered plants potentially that are growing in an agricultural sense. So beginning to grow fields of engineered organisms. And on the bottom right-hand side, we can see this is all the arable cropland on the planet. So if we're thinking about what are organisms that we want to engineer that could have a large scale impact potentially on our planet, on our society, arguably plants are a very attractive system to begin to start engineering. And so the overarching question my group's very interested in addressing is, can we expand the use and application of agriculture with synthetic biology? When we think of agriculture, we normally think of plants producing food. At the Joint Bioenergy Institute, again, we're interested in how plant biomass specifically can get broken down into its sugar monomers, so it could be fed to microbes to make other compounds like biofuels or bioproducts. But at the end of the day, what we're doing is manipulating metabolism. And so if you have this ability to engineer metabolism, you can actually go after many other traits. For example, potentially you could engineer new biomaterials or alter the properties of biomaterials that are derived from plants, such as lumber, cotton, or paper. Potentially, we could engineer new bioproducts that could be produced directly from plants, or pharmaceutical drugs, or even imbue new agricultural traits in plants. And so today I'm going to talk about 4 different areas that my group's been really interested in for a while. One, if we're going to do this complex engineering plants, we need the tools to be able to do this. And 2, really, if we want to engineer new products in plants, how do we begin to understand which products we should be going after? Thirdly, how should we be able to use synthetic biology to try to innovate on top of nature to make entirely new compounds? And finally, what are the implications of these methodologies on photosynthesis? So my computer fan is going nuts right now. I don't know if that's bugging you guys. Just holler if it gets too loud.
No, we can't hear it. Don't worry too much.
Okay, perfect. Sounds good. Okay, so really, if we want to do this sort of complex engineering, we need the tools to do so. On the bottom left, you can see some of the really complex and great engineering that we can do in things like E. coli. You can introduce many, many genes, control them with synthetic promoters, titrate their expression levels. And really, we just have not had the ability to do anything like this in plants. So kind of nuts and bolts in the very beginning of this talk, I'm going to go over some of the tools that we've been developing over the last several years so that we can go after more fun metabolic engineering. And so really, if you want to be able to go after complex traits, you have to be able to many times engineer many genes simultaneously, or introduce many genes simultaneously, or this in the plant world, we call this gene stacking. And so we've been able to take a page out of the yeast community's playbook, where they've been able to use the in vivo homologous recombination of yeast to actually stitch together large fragments of DNA to then, in this case, in the bottom right, build synthetic genomes. But in our case, we want to be able to introduce many genes simultaneously into a plant. We've been able to do this where we modify a plant transformation vector so that we can introduce multiple cassettes, many genes into a plant simultaneously. Here's just an example where we can introduce 3 different reporters, a GUS, a GFP, and an RFP into some soybean roots. But with this, we can go after more interesting traits other than just reporters, and actually begin to muck around with plant metabolism and produce things like, in this case, biodiesel called bisabolane, where you can introduce multiple genes, actually add genes upstream in the metabolic pathway to redirect metabolic flux and increase the production of this molecule bisabolane in a plant leaf. Similarly, we can go for complex metabolic pathways. Here we're going after the indole alkaloid biolacin, which is an anti-cancer compound, introduce all 5 enzymes that are required to make this and produce them also in a leaf. And so this is, the limiting step doesn't become actually introducing multiple genes. It then becomes, how do we coordinate the expression of all of these genes simultaneously in a plant? And so we've been really interested in how we can begin to design and build synthetic promoters to enable the targeted expression of transgenes. And some of the reasons we're interested in using synthetic promoters in plants is because, one, a notoriously hard problem to get over in plants is the problem of gene silencing. So when we express, say even just 2 genes with the same exact promoter, many times in a plant, these genes will then get silenced by the host. So on the right, you can see on the very right, the sickly looking plant, which is just a plant expressing nitrate reductase with a very strong promoter, the 35S promoter. And the plant already perceives this as a threat and begins to silence it. And so we believe that if you can build synthetic promoters that are avoiding similar cis elements or just promoter elements in general, you might be able to avoid the repetition of these sequences to avoid gene silencing. The other reason we're interested in synthetic promoters is many times we want to control gene expression levels. So say we're introducing a linear metabolic pathway into a plant. Say one of these intermediates is toxic to the plant. If we have the ability to tune gene expression and tune down the expression of the upstream enzyme and turn up the expression of the downstream enzyme, we can avoid accumulation of these toxic intermediates. And finally, plants are very different from unicellular microbes. They're multicellular. And really, a lot of the traits that we want to go after necessitate us to facilitate tissue-specific gene expression. And so we want to build a system where we can introduce multiple genes and deploy these genes in this tissue-specific manner. And so where we started off with this project was just to look through the lit and draw inspiration from nature. And arguably, one of the best transcription factors that has never been characterized is the GAL4 system from yeast. GAL4 has its galregulon. This transcription factor then binds all the downstream enzymes involved in galactose metabolism in yeast. And so what we wanted to do was to start building synthetic transcription factors that were based on other DNA binding domains. First, we went for GAL4 and start building these synthetic transcription factors, or in this case, we'll call them synthetic activators. We started building synthetic promoters by taking cis elements or transcription factor binding sites, in this case from GAL4, and fusing them to various minimal promoters. And we also were able to mine various genome sequences of various relatives of yeast that have also variation in the binding sites for these GAL4-TF binding sites. So from here, we just brute force built a giant synthetic promoter library, and we could characterize the expression strength of these promoters by expressing the synthetic activator. They would turn on a synthetic promoter to drive GFP. We have a constitutive promoter driving RFP so that we can normalize the expression level. And we just started expressing these in plant leaves. And here we can demonstrate that this was great. All of a sudden, we had this library of high, medium, low expressing synthetic promoters that we could deploy in plants. Now, remember, the reason we got interested in trying to build synthetic promoters was because we wanted to coordinate the expression of multiple genes simultaneously in plants in a tissue-specific manner. And so within this framework, with the synthetic transcription factor, synthetic promoter system, you can imagine using something like, say, a tissue-specific promoter to drive the synthetic transcription factor. This synthetic transcription factor can then control the expression of multiple downstream genes, all being expressed by synthetic promoters. So when we use, say, a seed-specific promoter to drive the system, we can see all of our reporters light up in the seed, and we see nothing light up in the vegetative tissue. We can show that the system is dynamically responsive to the environment as well. So say we swap out the seed-specific promoter for a phosphate-inducible promoter. When we add phosphate into the media, we don't see anything on. When we take away phosphate from the media, all the reporters light up. And so we think that this is a really nice system to begin to start deploying the stacking of genes so that you can coordinate the expression of many genes simultaneously in this tissue-specific manner. And so where we're moving towards is hopefully the rational design of promoters in plants. And so very simply, again, the way that we've been building these promoters are on two different levels. One of them is looking at essentially the cis element strength, and the other element is the minimal promoter strength. And so by fusing these two pieces together, you can imagine if you had the weakest cis element fused to the weakest minimal promoter, you'd be in this top left quadrant, and you'd have the weakest expression. If you took the strongest cis element with the strongest minimal promoter, you'd expect the strongest promoter strength in the bottom right. And if you made all the combinations of these, you'd expect a gradient from the top left to the bottom right. So we went ahead and did this experiment. We ranked our various cis elements and minimal promoters that we've been using, and we synthesized all these different variants. And we showed that we could recapitulate that gradient from the top left to the bottom right. Here we can cherry pick the 5 promoters in this diagonal and express them in a leaf. And you can see we have this gradual increase in GFP expression, which is exactly what we expected. And so everything that I've talked about so far is gene activation, turning on genes using these synthetic activators. But with the system, we can also introduce repressor logic, where we can just take that same synthetic transcription factor, slap on a repressor domain, and actually begin to start characterizing these repressors. So everything above this gray dashed line is an activator. Everything below, we're introducing a repressor into the system. So you can see here, when we add a repressor, we can see this drop in GFP expression, which is what we expect. We can normalize it to the RFP channel so that we can normalize and quantify how much transcriptional repression is actually happening. And so we think that this system could be really useful, say when you are trying to express, say, a given metabolic pathway in all tissue types except a given tissue type. So say we express the repressor in just the shoots, we express the activators constitutively, and therefore you'll only see expression in just the roots of the plant. And so this is just summarizing. A good chunk of the lab is really devoted to trying to build these synthetic biology tools so that we can do some pretty basic engineering in plants. We think that hopefully these tools will help along with our plant biologists, where we can begin to start reimagining and redesigning aspects of plant metabolism and plant physiology. So I just want to highlight that there have been like a lot of lofty ideas that have floated around the literature for many decades if you had this ability to engineer plants. But the real limitation has arguably been the tools to enable these sorts of projects. So say you want to introduce nitrogenase into plants. You probably want to express them in just the plant roots. Nitrogenase requires many different subunits, many different genes to be expressed in this very tissue-specific manner. So hopefully these tools could be helpful for those sorts of projects. If you want to modify aspects of photosynthesis, you want to target just the photosynthetic tissue. Or if you want to modify aspects of the grain, you again want to be able to target and coordinate the expression of genes in just those tissue types as well. And so we have this grand goal of trying to engineer plant metabolism and trying to ask this question of, what if you could engineer a plant to make whatever compound? And the real big question becomes, what compound should we be producing in plants? And so from the Department of Energy's perspective, from a bioenergy perspective, from a climate perspective, we've been very interested in how we can make biofuels economically feasible. And this kind of has meshed in with this broader question of, if we can, at what price point do we need to be able to make products in plants so that they are economically feasible? So intrinsically, it's just challenging to make biofuels cost competitive with petroleum-based fuels. So it's always going to be easier to literally pump out a product out of the ground than to try to make it. And when we talk about making biofuels, this is a really complex process where you take plant biomass, which is a very molecularly, physically complex material, break it down into its various components so that we can feed it to a microbe to then make it into a bioproduct. In this case, we'll be talking about ethanol. And so you can imagine with all of these steps, there are a lot of costs involved, which is why inherently it's been challenging to just make biofuels cost competitive with petroleum-based fuels. So our group has been approaching this problem a different way. Instead of trying to improve all these steps to make a biofuel, we're really interested in seeing how we can increase the value of the plant material from the get-go by making some sort of co-product, some sort of value-adding product. And if by doing so, maybe we can essentially subsidize the cost of making biofuels in general and make this cost competitive petroleum-based fuels. And so when we started off on this project, we just asked a really naive basic question: what molecules do we already extract from plants that are in the marketplace? And so there's this well-known inverse correlation between products that are high volume and their value. And so on one end of the spectrum, you have things that are like fuels and commodity chemicals, which we use a ton of in the market, and therefore they're very cheap. On the other end of the spectrum, we have things like pharmaceutical drugs or biologics, which we don't have very much in the marketplace, and therefore they're extremely expensive. So by looking at the lit, we could start looking for actual plant small molecules that are being sold in the economy. And here we can have your commodity chemicals, which are pretty cheap, your or boutique small molecules like antimalarial drugs, CBD, and then your extremely expensive anti-cancer drugs like vinblastine. And so from this, we had some targets that we could actually begin to put into a techno-economic analysis to try to guide our understanding and our efforts on what molecules should we be considering to engineer into plants. So by doing this techno-economic analysis, we have to envision what a biorefinery plant might look like. So here, what we're imagining is a bioenergy feedstock crop or a plant that has been engineered to produce a value-adding product. This would then be treated. The plant biomass would be broken down, like I told you, to make biofuels. So that plant material gets converted to sugars, it gets fermented, so you get bioethanol out of it. But the real difference in this biorefinery plant is we're adding this extraction step where in theory, we're taking out this value added product that will help subsidize the cost of making biofuels in general. And so from these techno-economic analyses, this is all just in silico, this is all theoretical. You can imagine very intuitively. Okay, so sorry, I need to break this down. So currently, again, because it's so expensive to make biofuels, the lowest that we can bring down the price of ethanol is $3.61 per gallon, this black bottom line. The Department of Energy for a long time has really challenged us to try to get ethanol down to $2.50 per gallon, but it's been pretty much impossible to do that. And so in these 4 different plots, we're imagining plants that are accumulating these specific value-added products. And so intuitively on the x-axis, you can see as you're accumulating more product, you're going to be dropping the price of ethanol because you're subsidizing the cost of making it. And so this can give us essentially a framework to begin to understand, oh, what concentration do you need to start accumulating these products so that you can, make a dent and make biofuels economically competitive with petroleum-based fuels. And so the billion-dollar question becomes, okay, can we actually make plants that are engineered to accumulate these compounds at these concentrations? And so what we were really quite shocked to see is that many of these plants are already producing compounds at dramatically higher levels than what would be needed to hit that $2.50 per gallon selling price. This is the anti-malarial drug artemisinin. It's produced by the plant Artemisia annua. And you can see here that many different Artemisia already accumulate much higher levels needed than to hit that price point. What's even more interesting is this pathway has been engineered in tobacco in the past. And so we know that we can actually accumulate levels that are in the ballpark of what are needed when shuttling this metabolic pathway into a heterologous plant host. Just to show that I'm not cherry picking here, we can go for other compounds like polyhydroxybutyrate, which is a bioplastic. This has been engineered into many different plants. And again, we're in the ballpark of what would be needed, if not accumulating higher levels than what would be needed to actually hit that selling price. And so I know these last few slides haven't been about biology, but I think it's been really important to do these sorts of analyses because if we're going to really strongly consider engineering plants for accumulating certain compounds, we need to see if this has a prayer of ever being economically feasible. And so we have that framework now to begin to ask, okay, if we are trying to engineer this compound into a plant, what concentrations do we need to hit to make this economically feasible? And this has been a tool that's been really helpful for guiding a lot of plant metabolic engineers in the field. Okay, so now we've talked a bit about what products should we be considering to engineer in plants. Because we're a synthetic biology lab, we're very interested in how we can try to make new compounds or innovate on top of nature in general to make entirely new natural products. So I would argue in the early days of synthetic biology, the focus was really on how you could take a known metabolic pathway and heterologously express it in a new host. But with synthetic biology, we have the ability to redesign metabolism in a number of different ways. And so we've been interested in how you could not just shuffle one pathway that already exists in nature into an organism, but actually begin to start mixing and matching enzymes to make new compounds that we've never seen before. So plants make a bunch of natural products to protect themselves from pests. We've been really interested in brassica vegetables. Here are some examples. Hopefully they're familiar to you because you're eating them on a daily basis, because they're actually really good for you. They're chock full of these indole alkaloids that we've been very interested in for a while. It's known that these are the compounds that protect plants from various pests and pathogens. And farmers have known about these molecules indirectly for a very long time. It's been known that when you actually till these brassica or cruciferous plants back into the soil, when you plant new crops on those soil, you decrease the disease incidence in those future crops. And it's thought to be because you're introducing these small molecules or these natural products into the soil that will protect future crops. So on top of being good for plants, they're also good for humans as well. We've been really interested in these two molecules right here, brasinin and cyclobrasinin. They've been very well studied by the pharmacology field for their anti-cancer properties. So we thought that that would be a really good starting place for trying to do some metabolic engineering. So again, because we've been really interested in trying to do kind of more synthetic biology to make synthetic metabolic pathways, we wanted to take this plug and play approach to try to design novel non-natural products or new to nature compounds. And so here in gray is the canonical biosynthetic pathway to make brasinin. I'm glossing over a lot of the enzymes. There's over 7 enzymes that are required to make this molecule, starting from tryptophan. And the only enzyme I want to highlight here is the first committed step, which is the cytochrome P450, the CYP79 that converts an amino acid to its corresponding aldoxy. And so the goal of this project was really to see if we could pipe in alternative substrates, alternative amino acid substrates into this pathway to make non-natural products that we've never seen before. And so we could swap out other P450s or CYP79s from other plants that we know that can accept other amino acids. So here we could borrow an enzyme from the plant Soteria that accepts phenylalanine instead of tryptophan. And if it went through this pathway, it would make this new compound that we're calling benzyl crucifixin. Now we're using the term crucifixin because this class of compounds has never been seen before in nature. We had to come up with a catch-all term. Because this pathway was inspired by cruciferous plants, we've been calling them crucifixins. Similarly, we can borrow another enzyme from a different plant, sorghum, that accepts tyrosine, and steal that CYP79 to make a hydroxybenzyl crucifixin, or borrow some enzymes from lotus to accept isoleucine to again make methylpropyl crucifixin. And so we can take all of these genes and express them in a tobacco leaf. And so here we can demonstrate in that tobacco leaf, we can make the native compound brasinin, which was great. But when we started to reconstitute our synthetic metabolic pathways, we were excited to see that, yes, we could make all of these new non-natural compounds that had never been described before. Here, we've synthesized the standard, so we're 100% sure that we're making these compounds. And so this was great. We could demonstrate that we could expand the repertoire of small molecules that could be produced in a plant. But what would be even cooler is if we could demonstrate that some of these new compounds had some sort of novel bioactive property that's never been shown before. So I'm going to back up a bit and mention that the native molecule brasinin is actually used as an antifungal compound to protect its native plant bok choy from various pathogenic fungi. And so we thought that would be a natural place to start, is to test the antifungal properties of our new compounds. And so the way we do this is we do a fungal growth inhibitory assay. So what we do is we take a bit of a fungal pathogen, we dab it on an agar plate, and here you can see in the picture, it grows out radially. Now we can challenge the fungal growth by putting in certain concentrations of our, in this case, our natural molecule, brasinin. And you can see here that the fungal pathogen doesn't grow as much because there's low-level inhibitory properties of our natural molecule brasinin. Now we can hit it with the same concentration of our new molecule that's never been seen before in nature, benzyl crucifixin, and we can see here that it doesn't grow at all. So we can be a bit more quantitative about this and actually measure IC50s. Here we're looking at the pathogen Alternaria, which is a major plant pathogen. It's a necrotrophic fungus. And when we measure these IC50 curves, we can very clearly see here that our benzyl crucifixin, our new molecule, dramatically outperforms the native molecule brasinin. So up until this point, what I've described is essentially just making a bunch of new molecules just to see if we can make new molecules. And then after the fact screen and see if they had some sort of novel active property. But we know from the pharmacology field that if we could introduce new actual moieties to these small molecules in a targeted manner, like specific moieties like halogenation, we could increase their efficacy against cancer cell lines. So here, by brominating brasinin, that's been demonstrated to improve the effects of brasinin. We can actually engineer halogenases from bacteria into plants to halogenate the 5 or 7 position of the indole ring of tryptophan. We also know that variants of brasinin, like cyclobrasinin, also improve their efficacy against cancer cell lines. So if we reconstitute this entire thing together, here we can already make the molecule brasinin. We know the last P450 required to make cyclobrasinin, and we can engineer these bacterial halogenases. In theory, we can make all these new chlorinated and brominated versions of both brasinin and cyclobrasinin. Of course, I wouldn't be telling you this unless it worked. And so here's the data when we reconstitute these pathways in a plant, demonstrating that, yes, we can make all of these new variants, halogenated variants of both brasinin and cyclobrasinin. And so now that we have, we could demonstrate we can make all these new halogenated versions of these compounds, we again wanted to revisit their potentially antifungal properties. Here we're adding another major plant pathogen, botrytis, into the mix. And when we measure their IC50s, we can see here that when going up against botrytis, this fungal pathogen, chlorobrasinin, this new halogenated version, is by far the most potent antifungal agent. Whereas when we look at a different pathogen, Alternaria, that benzoyl cruciferoxin molecule from earlier is actually the best. And so this paints this picture where if we increase the arsenal of small molecules we can make in a plant, we can actually tailor specific small molecules as biopesticides tailored to protect against various pests and pathogens. So again, none of this matters if we don't actually benchmark it. And here we're benchmarking IC50s against a synthetic pesticide that's actually sprayed in fields, pyromethanol. And so the natural molecule brasinin is not as good as the synthetic pesticide. Benzoyl cruciferoxin, it's better, but not as good. But our halogenated chlorobrasinin is pretty much on par with the synthetic pesticide. But again, none of this matters. If you can't accumulate enough of these biopesticides in the plant, it really won't matter. And when we look at the concentrations that are accumulated in our engineered plants, we can actually see here we're in the ballpark of what would be needed. And so the big picture goal is to see how far we can expand the metabolic repertoire of plants. Today I've been talking about this one molecule, brasinin, in the bottom left, but there's many other natural products that are produced from plants that humans are very interested in trying to get access to. We don't want to stop there. We don't want to just make these molecules. We want to see if we can use synthetic biology to try to innovate on top of nature and make new derivatives that might have enhanced bioactive properties. So I'm not going to, that's the only, metabolic engineering story I'm going to talk about, but I do want to mention that using metabolic engineering and synthetic biology, we can engineer many new traits into crops. Again, we talked a bit about how these cruciferoxins can be used as biopesticides, as agricultural traits. We've also recently been very interested in how you could boost the production of specific pharmaceutical drugs or chemopreventative compounds like glucoraphanin, which is why many people eat broccoli sprouts. We've been very interested in how you can actually make bioproducts and different bioplastics directly in plants, as well as trying to alter the properties of plant cell walls and biomass and biomaterials in general by introducing new small molecules that can either help the deconstruction process of biomass to make biofuels or alter the properties of these biomaterials like lumber. And so I do want to mention very briefly that all of this carbon is coming from photosynthesis through the enzyme rubisco, which Martin gave a great talk and summary on already. And so in this very last section, a good chunk of the group focuses on rubisco evolution actually, but I thought at the end of this talk, it might be good to not focus on rubisco because Martin was, probably was going to talk a lot about it and did talk a lot about it. But more from a synthetic biology standpoint, how can we begin to engineer new synthetic metabolic pathways to try to optimize photosynthesis? And so again, rubisco is the rate limiting step in photosynthesis. Again, it's the key enzyme in the Calvin-Benson cycle, which normally we think of as a carboxylase enzyme that fixes CO2. But every now and then it fixes O2 instead, which makes this toxic intermediate 2-phosphoglycolate, which then has to be recycled through photorespiration, which is this energetically costly pathway that results in the net loss of a carbon and a nitrogen molecule, which is why things like the pyrenoid exist, so that you can avoid this costly reaction. Now, there are a lot of people in the synthetic biology community that are really interested in ditching rubisco altogether and trying to figure out how you can build synthetic carbon fixation pathways from scratch. And the reason we think that you can do this is because we know that the Calvin-Benson cycle is not the only carbon fixation pathway in nature. There are actually 6 other carbon fixation pathways in nature other than the Calvin-Benson cycle. But we're not even limited to that. Kind of similar to mixing and matching enzymes to build the synthetic pathways to make those cruciferous enzymes I talked about earlier. One can also mix and match enzymes to build these synthetic carbon fixation pathways that don't use Rubisco and avoid kind of the detrimental aspects or the non-advantageous aspects of Rubisco. And so these kind of building these synthetic metabolic pathways has been used to address, again, this problem of photorespiration, where people have tried to engineer these synthetic shortcuts to try to avoid all the costs of photorespiration by introducing these synthetic metabolic pathways into plants. So we've been really interested in this space where one of those naturally occurring carbon fixation pathways is called the 3-hydroxypropionate bicycle, found in this really odd bacteria called Chloroflexus. And we got interested in this pathway because if you could actually steal this half of the pathway, plug it into a phototroph like a cyanobacteria, it would actually work as a synthetic photorespiratory bypass, where if Rubisco fixes O2, you would then make 2-phosphoglycolate to glyoxylate, which would then go into the synthetic pathway to kick out pyruvate and get recycled through acetyl-CoA carboxylates, which actually fixes more CO2 in the form of bicarbonate. And so we like this pathway because it results in no loss of nitrogen and actually a gain in carbon compared to other synthetic pathways that have been going after improving carbon fixation in plants. And so when we've been able to engineer this pathway into cyanobacteria in the past, we can, again, stack all these genes together, introduce them into cyanobacteria, and demonstrate that, yes, we have activity of all of these enzymes in cyanobacteria. And really, it stopped there because ultimately we want to move this into a plant, but we really needed to have the synthetic biology tools to express all these enzymes in a coordinated manner. And now that we've done that, that's something that my group's very interested in moving towards. And so in summary, my group's been really interested in how we can build the basic synthetic biology tools to carry out complex engineering. We've been really interested in exploring what products we should be making in crops, and also how we can try to use synthetic biology to make new compounds not seen before in nature, and more broadly, how the implications on metabolic engineering has on photosynthesis. So this is just my last slide. Really, the big picture is we want to use plants as a scalable and sustainable platform for synthetic biology. Plants are the foundation of any bio-based economy. Right now, we use so much petroleum. There's so much interest in trying to wean our society off of petroleum. And we get so much carbon out of the ground. If we're going to go cold turkey on it, when we look at places where we can actually get that amount of carbon, agriculture and plants are pretty much the only place where we can get that amount of carbon to replace petroleum. And so we think that plants are a nice photosynthetic platform to also make a wide range of compounds. And we think that deploying synthetic biology on these agricultural scales will really open up new opportunities in general. And so with that, this is the group, this is the funding. The cruciferous engineering was a collaboration with Beth Satley and Jay Kiesling. The techno-economic analysis was a collaboration with Kern Scone at JBay. And with that, I'd be happy to take any questions.